Okay, I think we gave people some time to get in here. So we've already had a quite active pre-talk discussion, but we're really looking forward to hearing more about what you have to say, connecting your in vitro systems to in vivo brain phenotypes in many ways here. So thanks for coming, Jason. We've been aware of your work for a long time and very happy that you could come share it with us today. Thanks a lot.
Thanks so much. Thanks, Carlo. Okay, so excuse me. Today I'm going to talk to you about modeling genetics altering human cortical structure. So today I'm going to focus on the neocortex, which is the outer covering of the brain, which is greatly expanded in humans relative to other species. So specifically, the surface area of the cortex is over 1,000-fold expanded compared to mouse. And it's thought that human enhanced cognitive and social abilities are possible in large part because of the expansion of the cortex. In addition, alterations in the development and structure of the neocortex are found in individuals with psychiatric disorders, motivating their use for studying these disorders. So genetics is clearly one large part of what has led to the expansion of the cortex and what causes it to be altered in individuals diagnosed with psychiatric disorders. So in general, how do we find genes that impact the structure of the cortex? Well, there's a large body of research identifying genes impacting cortical structure and development. In humans, this body of research is mainly identifying rare genetic variants that have very large effects on brain structure, usually microcephaly, very small brain, or macrocephaly, very big brain. This is an example of a translocation breakpoint leading to microcephaly near a gene called EOMS or TBR2. And we usually find these genes in human populations, but then create models in mice to understand what their mechanistic function is. So in those mouse models, for example, you can take your wild type mouse and knock out the gene EOMs and see somewhat of a similar phenotype there. So this approach has been very successful. We've identified many genes associated with microcephaly and macrocephaly. However, this noticeably excludes many other types of genetic variation that impact brain structure. Specifically, what we're going to focus on here today is how common genetic variants impact the structure of the brain. So in order to find common genetic variants associated with inter-individual differences in brain structure, we need to first get large populations of individuals who have undergone genotyping and MRI to take basically pictures of the brain. The reason we need these large populations of individuals for this approach is because we expect that common genetic variants will only have a small effect on complex phenotypes like brain structure. And large sample sizes provide power to detect these small effects. So in these types of studies, we can identify specific genetic variants that are associated with inter-individual differences in human cortical structure or the structure of other brain regions. And in my toy little example here, you can see that the A allele is associated with a bigger brain, while the C allele is associated with a smaller brain. So genetic associations, though, once we identify them, are just kind of the first stop in a pathway to understand the mechanisms associated with differences in brain structure. So our lab aims to find genetic variants associated with different brain structure traits and also explain their molecular mechanisms. The way we do this generally is to use populations of individuals with inherent genetic variation and determine whether genetic variation is associated with differences in each of these different phenotypes. So these type of phenotypes that we're interested in are, for example, gene regulation, molecular switches like enhancers or promoters that turn on or off gene expression, the levels of gene expression themselves, including within specific cell types or specific conditions, mechanisms like proliferation, and then ultimately leading to differences in macroscale brain structure. So today I'm going to go over some of our findings at these different levels to give you an idea of how we are trying to link genetic variation to each of these different traits with the aim of understanding the mechanisms leading to differences in cortical structure and ultimately risk for psychiatric disorders. So first, I'm going to give you some background in how we identified genetic variants influencing the structure of the human cortex. So we can measure the structure of the cortex using magnetic resonance imaging, MRIs. And in MRIs, we can segment out the cortical ribbon using automated tools. And you can see that colored here on this brain image. We can do this both for individual cortical gyri and sulci, which are marked by these different colors. And we can measure different features of the cortex, for example, the thickness or the surface area of the cortex. At this point, this can be done in really impressive sample sizes by combining data sets from all across the world. And when I was a graduate student, I co-founded the Enhancing Neuroimaging Genetics Through Meta-Analysis, the ENIGMA Consortium, with the goal of identifying genetic variants influencing brain structure. So I'm going to go over one of our recent studies, which identified genetic variants influencing cortical structure, where GWAS was conducted in around 34,000 individuals with replication in an additional 17,000 individuals. So this study design allowed us to identify the genetic variants contributing to inter-individual differences in brain structure. So these are the results from our study. So our study was the first to identify the genetic variants affecting cortical structure in a really well-powered way. So this is an ideogram showing each of the different chromosomes, and marked are all the different loci that have been associated with inter-individual differences in cortical structure. The colors here represent different regions of the cortex, and the symbols represent either different significance thresholds or different phenotypes. So what did we learn from this? Well, first, we identified common variants impacting cortical structure. So we were able to find genetic variants impacting the structure of the human cortex that were not these rare microcephaly or macrocephaly associated mutations. These common variants impact both global measures, like the overall size of the cortex, as well as individual regions. We identified many more loci actually impacting surface area as compared to thickness. And often genetic variants impact just one cortical structure, as you can see, for example, here, but sometimes they impact multiple different cortical structures, as you can see in example here. Okay, so these are kind of guideposts that tell us something about what the genetics are that cause differences in the size of the brain. So what biology can we actually learn from understanding these individual genetic markers? So first, in our GWAS of cortical surface area, we found that common genetic variation associated with differences in cortical surface area was clustered near genes in the canonical Wnt pathway. We were excited about this because Wnt stimulation is known to increase neural progenitor proliferation and pattern the cortex. And this data kind of I think I lost you guys. Sorry.
No
Can
worries.
you?
I think, yeah, we got you back now.
Okay. I'm going
Don't
to
know
share
what
this again. Sorry about that.
No worries.
Can you see the slides?
Yes,
Okay,
but
great.
I think it
Sorry,
advanced since last time I saw.
Jason,
Yeah, yeah.
can you please go a couple of slides back?
Yeah.
Yeah,
Oh,
yeah,
sure.
yeah. No,
On
no,
Wnt.
the
We saw
next
the
one.
Wnt one. You were talking about
Yeah.
Wnt.
You saw the Wnt one.
Yeah, I
Okay,
lost
great.
you here. Yeah,
Okay,
thanks.
great. So
Thanks.
yeah, the summary of this slide was that the GWAS for individual regions associated with cortical structure was enriched near genes involved in Wnt stimulation. So we were excited about this because there's a known developmental pathway, like Wnt stimulation is a known developmental pathway involved in brain patterning and proliferation. And this kind of implies like an early developmental time period, like genetics having an effect in early developmental time period, leading to differences in adult cortical structure. Okay. So next, we wanted to determine if there are specific developmental time periods or cell types that contribute to differences in human brain structure. And we pursued this through what's called a partitioned heritability approach, which effectively asks if genetic association signal for cortical structure here is enriched in defined regions of the genome, for example, those that are annotated as active regulatory elements within different tissue types, like, for example, brain or liver. And here you can see in my little toy example that there's an enrichment of GWAS signal in brain active regulatory elements. So we pursued this partitioned heritability approach using different tissue types and just to summarize and different cell types and different stimulation states as well. For example, Wnt's specific regulatory elements here. We did this for both global thickness and global cortical surface area. And we found that there was an enrichment in heritability within regulatory elements present during fetal development, specifically in progenitor cells, and that regulatory elements in progenitor cells affected by Wnt signaling through these two different compounds also show enrichment in cortical surface heritability. So overall, what does this say? What did we learn from doing our GWAS of cortical surface area? We can try to kind of imply which cell types and developmental time periods influence cortical structure. So what is implicated is that the cell types that influence cortical structure are progenitor cells, which are these cell types that form all of the neurons in the cortex and are present early in fetal development from 8 to 24 post-conception weeks, and then lead to differences in these alterations in regulation of these cells lead to differences in size of the adult cortex later on in development. So this is probably, we think, human-derived evidence for an old hypothesis called the radial unit hypothesis. And the idea here is that genetic variation within regulatory elements impacts progenitor-associated genes before birth, presumably influencing their fate decisions, which then results in impacts on adult cortical surface area. So to explain this a little bit further, during these early periods of development, when progenitor cells exist, this is what a cartoon of what the developing cortical wall looks like, where we have a germinal zone containing progenitor cells that then differentiate into neurons that migrate up along radioglial fibers. These type of neural stem cells make fate decisions early in development. They can make symmetric fate decisions to form more neural progenitor cells, or make asymmetric fate decisions to form neurons. However, neurons are postmitotic, so once you form a neuron, you kind of have lost some of your pool. So if we have progenitor cells that make symmetric fate decisions to form progenitor cells, that make symmetric decisions to form progenitor cells, we can overall increase the size of the progenitor pool, leading to increased neuronal production, which then leads to increased cortical surface area. So this hypothesis has been provided to explain why humans have bigger cortical surface area than non-humans. But here we think that our evidence kind of leads to support for this radial unit hypothesis explaining inter-individual variation within the human population as well. Okay, so to summarize this first part, we identified common genetic variation influencing human cortical surface area and thickness. And we found that genetic variation influencing postnatal cortical surface area is enriched in active regulatory elements within the prenatal brain progenitor cells, which we think is human evidence for the radial unit hypothesis. And finally, associations near Wnt signaling genes are involved in brain structure, implicating this known developmental pathway in shaping the human brain. Okay, so as I said, though, we've discovered many loci impacting brain structure, but these are just the starting point on the pathway to explain mechanism. So these loci are in general kind of difficult to interpret, and if we take a genome-wide association signal locus that's associated with one particular brain region, in this case here, the lingual gyrus and the occipital cortex, we can see that it's actually pretty complicated. So here, as a location on the genome, we find that there's many different dots here, each which represents a SNP that are associated with differences in the size of the lingual gyrus, the cortical surface area of the lingual gyrus. We have some dots over our genome-wide significance threshold, so they're significantly associated with differences in the size of the lingual gyrus here. But none of these dots are actually changing the protein coding sequence of any of the genes around, so they're all non-coding. The causal variance within the locus is unknown due to linkage disequilibrium, the correlation of these nearby variants in the genome. So we don't know which of these variants, or maybe more than one of the variants, is actually having an effect. Because these are non-coding variants, the causal gene is also unknown, and genetic variants, though that they're the same in every cell type in the body, they don't have to have an effect in every cell type or in every context. So the cell types and developmental contexts that these variants exert their function in are also unknown. And finally, what the mechanism is that leads to specific regional effects, despite these variants again being present within all the cells of the brain, is also unknown. So, these are, again, a really good starting point, but there's a lot of mechanism to explain. So I'm going to go over some of how our lab is trying to explain those mechanisms. So because most of these variants lie in the non-coding genome, we often think that they have gene regulatory potential. That is, that genetic variants tune the switches that turn on or off gene expression. So how do they do that? Well, the hypothesis is that genetic variants can disrupt things like transcription factor binding, leading to differences in transcription factor binding to a specific location of the genome, and subsequently differences in chromatin accessibility, which is kind of a proxy for transcription factor binding. They can subsequently impact downstream gene expression and ultimately lead to differences in cellular behavior that lead to differences in the size of the cortex. So we can measure how genetic variation affects gene expression in a study design called an expression quantitative trait locus study, or EQTL study, where we get a population of individuals who are genotyped and measure their expression. We can do the same idea using chromatin accessibility in a population of donors to understand how genetic variation changes these regulatory switches here. And that's called a chromatin accessibility QTL study, or CAQTL study. If we find that the same genetic variants are associated with differences in chromatin accessibility, gene expression, and ultimately macroscale brain structure, we can assume we have explained some aspect of how this genetic variant leads to risk for differences in brain structure. Okay, so this approach has some advantages. For example, it models endogenous genome function, but also there are some disadvantages as well. There's still confounding by LD and background genetics. It's not just that there's one location in the genome with variation, there's many locations in the genome with variation. We do our best statistically to control for that, but this is still a possible confounder. So, despite the genetic variants will be constant across all cells in the body of an individual, the variants may exert their function only in certain cell types or cell states, meaning that QTLs can both have cell type and context specificity. So cell type specificity may be driven by transcription factors that are only expressed in some cells, but not other cells, leading to differences in allelic effects only in some cells. They may also be driven by context specificity, where in the absence of a stimulus, a transcription factor does not translocate into the nucleus. But in the presence of a stimulus, a transcription factor can translocate into the nucleus and lead to differences, allelic effects, and differences in gene expression. So I'm going to talk to you about how we're assessing both cell type and context specific QTLs. So to do this, we used a cell culture system for evaluating our context and cell type specificity. So when I was a postdoc in Dan Geschwin's lab, we acquired human fetal brain tissue from over 90 different donors. And then we cultured those cells and cryopreserved them, transferred them over to UNC, and we began these large-scale differentiations to understand how genetic differences across all these donors lead to differences in things like chromatin accessibility or gene expression. So we profiled these donors both at two different cell types, neural progenitors, which are SOX2 positive, PAX6 positive radioglia, and we differentiated them out for 8 weeks, labeled with a virus that specifically expresses GFP in neurons, sorted out the neurons, and performed chromatin accessibility through ATAC-seq in our neurons and progenitors, as well as RNA-seq in our neurons and progenitors. So doing this type of study, we were able to discover thousands of locations where genetic variation is associated with differences in chromatin accessibility or brain structure using this QTL study design. And I'm going to selectively pick one for you to go over. So here's an example of a global surface area, a GWAS for global surface area. So genetic variants associated with differences in the surface area of the human cortex. And you can see that these are, again, genetic variants, but they're a whole bunch of variants. They're high LD. We don't really know which is the causal variant or which is the causal gene, and none are changing the protein coding sequence of any of the genes in the region. We can layer on our information that we discovered for progenitor EQTLs or neuron EQTLs. And we discovered that, in fact, there's a co-localization between our progenitor EQTL for CENPW and cortical surface area. So what this suggests is that genetic variation working in progenitors is altering the function of the expression of CENPW and probably ultimately leading to differences in cortical surface area. We also layered on our CAQTL evidence in this region So, and we can zoom further into a particular chromatin accessibility peak affected by colocalized genetic variation. So here's that peak showing differences across genotype and an individual SNP within the complicated LD region that we can suggest as causal right at the promoter region of CENPW. So using this combination of data, we can identify an individual SNP within an individual cell type that regulates a particular gene, CENPW, associated with differences in cortical surface area. So the idea of this is that we can use inherent genetic variation in a population of neural progenitors that alters chromatin accessibility and gene expression during human neocortical differentiation. If we use these cell type specific chromatin accessibility QTLs, they can lead to fine mapping and regulatory mechanisms of both eQTLs and GWAS loci, and overall that genetic variation has impacts during neurogenesis that can influence adult brain structure phenotypes. So I quickly went over our cell type specific QTL work to focus on our newer stuff, which is on context specificity. So now I'm going to talk to you about how we've been using our cell culture system to study context or stimulus specificity. Okay, so here we're going to use a similar study design to previous, but instead of just culturing our cells in the absence of any stimulus, we're going to add both a vehicle and some stimulating condition to all of our different cells. These cells have inherent genetic variation that we've already genotyped, and then we measure gene expression or chromatin accessibility in all these cells. What we think this gives us is context-dependent eQTLs, which another way of calling this is that we're doing gene by environment interactions conducted in a dish, so G by E's in a dish. We think that this approach of doing G by E's in a dish has several advantages. First, it's difficult to do these type of assays in postmortem tissue, mainly because you can measure exposures in postmortem tissue, but those exposures are usually across a life and not really specific. Whereas here we have a living cell culture system that we can add exposures to. Unlike in postmortem tissue, here we can design it so that each genotype receives both the vehicle and the stimulus condition. And the dose or concentration of the stimulus is very well controlled, as are the cell culture environments. And we can do this with any population of primary or iPSC-derived cells. Of course, there are disadvantages to our G by E in a dish approach as compared to using postmortem tissue, for example, in vitro and vivo differences. And of course, we're using mainly cellular or molecular phenotypes because behavioral phenotypes, which are the ones we really care about, are difficult to evaluate in a dish. Okay, so our stimulation paradigm for doing G by E's in a dish, of course, we need to stimulate with a relevant pathway that's important for neural development and cortical structure. So we've been focusing on the canonical Wnt pathway for several reasons. So first, stimulation of the Wnt pathway is known to affect neural progenitor proliferation and patterning in the developing brain. Second, de novo mutations associated with autism have clustered in Wnt pathway genes, implicating these as important for psychiatric disorders. And third, I've already showed you some evidence that suggests that Wnt signaling is important for inter-individual differences in the size of the human cortex. So all of these indicate that genetic variation in neural progenitor cells may exhibit brain trait relevant functionality in the context of Wnt stimulation. So a quick primer on the Wnt pathway. So the canonical Wnt pathway has been very well studied and its signaling cascade is very well known. So in the absence of Wnt, at TCF left binding sites, Groucho is bound and the chromatin is closed. But in the presence of Wnt, beta-catenin can migrate into the nucleus, leading to the acetylation of Wnt of these TCF left binding sites and to opening of chromatin at TCF left binding sites. So with skipping a lot of parts of the Wnt pathway there, just to focus on the ones that are important for our study, we expect opening of TCF left binding sites during Wnt stimulation, whereas closing in the absence of Wnt stimulation. So how do we stimulate Wnt signaling? Well, we can add either the recombinant ligand, Wnt3a, which we can buy, or a molecule called CHEER that inhibits GSK3 beta and allows beta-catenin to translocate into the nucleus and lead to the opening of these TCF left binding sites. So we then conducted this G by E in a dish, so the stimulus-specific QTL, in our neural progenitor population from 82 different primary human neural progenitor cell lines. So in these primary human neuroprogenitor cell lines across all these 82 donors, we added either vehicle or Wnt3A or cheer. So these are our Wnt stimulators. And then we measured chromatin accessibility via ATAC-seq and gene expression via RNA-seq. And then we aimed to find genetic variants whose function was modulated by stimulation of Wnt signaling. So in the presence of Wnt signaling, we may see a genetic effect, whereas in the absence of Wnt signaling, we see no genetic effect. So these kind of Wnt-sensitive QTLs were what we were aiming to find. And this study was led by Nana Matoba in my lab, Brandon Lay, and Jordan Vallone, and was recently just published. So the first thing we wanted to do in this study was to find peaks that were opening or closing due to this Wnt stimulation. And we expected that those peaks that were opening due to Wnt stimulation would be enriched for motifs for these TCF-left binding sites. And indeed, that was found for both the cheer version of stimulation and the Wnt3A version of stimulation. So this result was expected because of what we know about the signaling cascade, but also it gave us much greater confidence in our downstream analyses that basically Wnt activates the downstream effectors of Wnt. So we were pleased to see that. And having satisfied ourselves that the assay did as we supposed, we then asked whether there was a general enrichment of brain trait GWAS heritability using that same partition heritability that I described previously with different psychiatric disorder brain traits in these Wnt responsive elements, these WREs here. And here you can see that for some developmental brain-related traits like ADHD, autism, schizophrenia, as well as cognitive traits like intelligence and educational attainment, we do see an enrichment of GWAS signal within these Wnt responsive regulatory elements. So I have also already showed you that this thickness and surface area version of this in an earlier slide. So what this all implies is that common variants within Wnt responsive genes and regulatory elements contributes to inter-individual differences in brain structure, psychiatric disorder risk, and cognitive ability. So genetic variation within these Wnt responsive elements is important for these downstream traits. So then we wanted to go on and identify specific genetic locations that were involved in regulating differences in gene expression or chromatin accessibility within the context of Wnt3A or cheer exposure as compared to vehicle shown here. And we identified many QTLs, so 43,000 chromatin accessibility QTLs. Those are those are CA SNP, CA peak pairs, so like pairing between the two. In each condition, regulating 36,000 unique CA peaks. We also identified 2,000 EQTLs in each condition, regulating around 3,000 unique E genes. So these are shown in these what are called Miami plots here, kind of showing a GWAS on two different sides for both the stimulation and the unstimulated. We also then looked for how genetic variation can be modulated by the Wnt stimulation, and all of those that survived an interaction significant test are shown by the circled dots here. So basically, using our approach, we identified thousands of context-dependent molecular QTLs, showing that genetic variation does have stimulus-dependent effects. So genetic variation can be modulated, the function of genetic variation can be modulated by a stimulus. So we can compare those QTLs across different conditions, shown here in an upsetter plot. And most QTLs that we detected, for example, here CA QTLs and here EQTLs, were consistent regardless of condition, regardless of stimulation condition. However, some of the QTLs were only detected in the stimulus conditions in either Wnt3A or cheer, and those are shown here. And overall, we get an increase in CA peaks, so those peaks that had a genetic variant associated with them. There was a 66% increase in CA peaks detected due to stimulation, and a 50% increase in E genes detected due to stimulation. So these results show that Wnt stimulation reveals context-dependent genetic effects on gene regulation that were previously undetected in unstimulated cells. We also explicitly tested for differences across conditions using two approaches. One is a Bayesian approach called MASH, and the other is an interaction test, which we call the RQTL. And we found basically thousands of CA peaks and over 100 E genes with statistically significant condition-dependent effects. So sometimes those interactions that were accentuating genetic effects that existed in both conditions, and those are shown like here. And sometimes those interaction effects were found specifically in one of the stimulated conditions, but not in the vehicle condition, as shown like here. Okay, and so to give an example of a stimulus-specific colocalization, so here we have a cheer responsive EQTL that was not detected in vehicle that colocalizes with a schizophrenia GWAS signal. So we have the schizophrenia GWAS signal, again, in a complicated region. We don't know a specific gene being affected, and that gene being affected was not revealed in the vehicle condition, was only revealed in the cheer stimulated condition. This EQTL was to a gene called ANKRD44, and this is a gene that's involved in proliferation, and it may indicate why it's involved in differences in hippocampal subfield as well, specifically the presubiculum body, as well as risk for schizophrenia. So overall, Wnt-specific QTLs allow us to better explain the mechanisms for GWAS, complicated GWAS traits like schizophrenia GWAS.
Jason, can I just ask you a question while you're speeding through this?
Sorry, yeah.
Yeah, so one of the things that's sort of bugging me as you go through this, is this all done on these 80 human progenitor lines?
Yes.
All these data. And so
Yes.
then when you make a statement about a specific brain region like this subiculum region, is the effect that you see, maybe you can formulate the question for me. Do you see what's bugging me? It seems
Sure.
like the cells must represent something fairly generic. And are they all behaving the same way? And then, some aspect of the phenotype appears to be quite specific. Is that helpful to ask that question?
I think that is fair. Yeah. So, our progenitor cells. So first, let's describe the progenitor cells. So these are, PAX6 positive, SOX2 positive. They stain somewhat for HOPX. They are basically radioglia. We did remove cells that had high. So we would get tissue when we isolated these cells, and that tissue is like all dissociated. We don't really have like the ability to specifically dissect the cortex. But the cortex looks kind of flat, and so we tried to select the cortical regions. Then we would threshold to remove any of the donors that had high expression for things like NKX 2.1. So we're pretty sure that we're getting a pretty solid population of radioglia from cortical regions. We don't have specificity to those cortical regions, though. So it's not like we're getting frontal cortical regions consistently or occipital cortical regions. Okay, so back to this slide you were asking about. So here we see a colocalization for our cheer responsive QTLs with hippocampal subfield. So this hippocampal subfield GWAS was conducted in like 30,000 individuals from the UK Biobank. So I guess your question is like, why do we see in cortical progenitors a Wnt responsive QTL that colocalizes with hippocampal subfield, even though these are cortical progenitors, not hippocampal progenitors? And, I think what you said kind of makes sense that, the idea would be that Wnt responsiveness, like the responsiveness to Wnt signaling, would lead to some kind of generalizable phenotype in progenitor cells that leads us that reveals the function of this genetic variant. Would it be better to have hippocampal progenitor cells and redo this type of study? Yes, I think that would be more convincing for sure, and would sort of give us more confidence that you could actually make these connections. I think that type of study in the future we can think about with iPSC studies versus these progenitor cells. These are kind of like fate committed to cortex, we think. So we don't really think that these are kind of going to be reverted to being hippocampal progenitor cells in this. So, yep. Does that make sense, Ron, or any?
Well, I think it's a very interesting issue. I'm amazed that you can get so much traction out of the model, right? And I think it seems to me it's suggesting that there's something. I mean, Kristen Brennan isn't on this call, but she gave the last talk in this series, or maybe she is on the call. But this comes up all the time in using cellular models for psychiatric disorders, that you get phenotypes without apparently having to get hugely specific about the cells you're using. Anyway, let's keep going. Thanks so much.
Okay, yeah, I think it's fair. And yeah, I think I'll show some other data that kind of lends to what you're saying, that, with human cell culture models, we're not getting things that are perfect models of human fetal tissue. So even with iPSC cultures, I'll show that too in just a second. So maybe it will spark more conversation. Okay, so I talked about doing those colocalizations. I showed one example of a colocalization with this ANCR-D44, but we did this for many different GWAS traits that we were able to gather together for many different brain traits. And so most of our CA peaks, as shown here, basically do not overlap with a GWAS peak, but there's a subset basically that do seem to have some downstream impact. And those can be, some of them are context dependent, meaning that their function is revealed really in the context of this Wnt signaling. And if we add Wnt signaling or, two different types of Wnt signaling, we get increased numbers of colocalizations. Basically, this suggests that as we sort of increase stimulation states, we're going to be better able to explain EQTLs or CAQTLs underlying risk for psychiatric disorders. Okay, so to summarize this portion, germline genetic variation is identical in all cells, but can have effects in certain contexts. And Wnt responsive expression or chromatin accessibility QTLs in neural progenitor cells can explain additional brain trait associated loci that are not detected in unstimulated states. And in general, we think that this cell culture-based gene by environment interaction may be generally useful for identifying genetic effects on cellular or molecular phenotypes after exposure to environmental stimuli, or a pharmacogenomics approach, prioritizing individuals who may best respond to a drug. So we are pursuing this in other projects in the lab currently, but now I'm going to switch over a bit to a different study. So in this previous study, I talked to you about QTLs using endogenous variation to explain differences in gene regulation through chromatin accessibility or gene expression through EQTLs. But of course, there were disadvantages to that approach. So an alternative approach to utilizing endogenous variation is to introduce DNA segments that we generate into the cell. And those DNA segments can contain alleles at a SNP that's already associated with cortical structure, as well as some surrounding sequence. Those alleles can then be tagged to random barcodes, and then we can use RNA sequencing to measure the expression of this construct using by evaluating the amount of the random barcode, the reads overlapping to the random barcode. So this type of study is called a multiplex parallel reporter assay, EMPRA. And these EMPRA types of studies have some advantages. They model individual variant function, allowing separation of background genetics and LD. So they overcome some of the limitations that exist in QTLs. However, they have their own limitations as well. For example, these have disadvantages like they are modeling exogenous function in the absence of important things that determine gene regulation, like chromatinization. So they have advantages and disadvantages, but we wanted to use this assay as well to understand these cortical structure associated SNPs. So we ran an EMPRA assay using, together with our colleagues, Nana Matoba in my lab, Jessica McAfee in Heijung Wan's lab, and Heijung herself. So we did this in primary human neural progenitor cells, and we selected SNPs that were already shown to be CAQTLs, so those regulating chromatin accessibility, as well as our Enigma GWAS from cortical structure. So for this Enigma GWAS, we selected over 9,000 SNPs associated with inter-individual variability in cortical structure. We selected those SNPs with p is less than 1 times 10 to the negative fifth, so some nominal association to cortical structure at genome-wide significant loci. Those SNPs were selected for having a genome-wide significant locus associated with some brain structure or global structure. But note that there are some biases that go into this data. For example, we had more genome-wide significant loci in occipital cortex than we did in frontal cortex. Also, the number of variants tested at each locus varied by the LD structure of that locus. So for very big LD structure loci, like the chromosome 17 inversion locus, we tested a lot of different variants there. So in total, we tested around 19,500 elements in this EMPERA study. So the way we designed this was that we cloned out these sequences that contained the SNP of interest and both alleles. We tagged each allele to around 30 different barcodes on average. Those barcodes were then cloned into a plasmid, and those plasmids were then transfected into primary human neural progenitor cells. Those primary human neural progenitor cells were subjected to either Wnt stimulation using cheer or vehicle for 48 hours. And after that, we measured gene expression using RNA-seq of the barcode, and we used DNA-seq to measure the overall plasmid count as a normalizing factor. We did this with both the Enigma and CAQTL data, as well as positive controls. These are promoters that are commonly used to drive large amounts of gene expression. So we expected high barcode counts for these. And negative controls, which were just 150 different scrambled sequences from the Enigma library. We then quantified the level of activity in these barcodes and compared them to negative controls to determine if the EMPERA elements were active, EMPERA active elements. We also looked for condition specificity of these EMPERA elements and for allelic effects of these EMPERA elements, both in the vehicle and the stimulation condition. So in this type of study, we're again trying to find the function of these genetic variants that are associated with differences in cortical structure. So first, we wanted to show if EMPERA has consistency across replicates. So we did this by we had 14 different replicates. They were all from the same donor, but different replicates, meaning different wells, in this case, of the donor. The replicate correlations were very high. So you can see an example replicate correlation here. And we found that these individual positive controls, so like EF1 alpha promoter or CMV promoter, had really high transcriptional rate, meaning high barcode count, well above negative controls. And our CAQTLs sometimes had active elements and sometimes did not. So we next sought to evaluate the concordance between our CAQTLs from our previous work and the EMPERA conducted in this same cell type. So both of these were conducted in primary human neural progenitor cells. And we specifically chose CAQTLs where the SNP was in the peak and the CAQTL was replicated across two separate differentiations. So these are very strong CAQTL results. But surprisingly, there basically was no relationship between EMPERA allelic fold changes and CAQTLs. So CAQTL allelic effects are generally not recapitulated in EMPERA. What we did find, though, that though there was limited concordance between EMPERA activity and allelic effects in CAQTLs versus EMPERA, the strongest predictor of detecting EMPERA allelic effects was showing EMPERA activity above levels compared to negative controls. So we did detect allelic effects when the EMPERA activity was active or above the negative controls. So this is actually kind of somewhat consistent with current approaches in the EMPERA fields to do what are called expression modulating variants or MVARs. And so for all subsequent analyses, we only evaluated allelic effects when there was a significantly active above negative control EMPERA element. Okay, so now moving on to the Enigma library. So the Enigma part of this EMPERA library. We were trying to identify cortical size related regulatory variants. And we tested the regulatory activity of these elements and found 988 of our study of around 9,000 variants that had significant EMPERA activity. So these 988 variants were pretty well distributed across different loci, where around 70% of different loci contained at least one EMPERA active element. Now, most variants were not EMPERA active, though. Only around 7% of variants were EMPERA active. So, but since a locus contains multiple variants, around 70% of loci were had some EMPERA active element in them. We also found that in general, these EMPERA activity was shared between our vehicle and Wnt stimulated condition, although there were cases where we identified genetic variants or loci that only had activity in the vehicle or the stimulation condition here. So we also tested using interaction tests to see if there were significantly different effects in the conditions, like in the stimulated condition, and did identify a small subset of those as well. So overall, most loci contain some EMPRA active elements. Okay. So in order to determine the downstream impacts of these condition-specific EMPRA elements, we then mapped each of these elements to genes they could possibly function on using either location at the promoter or chromatin interactions to genes of interest for distal elements. And interestingly, EMPRA elements with higher activity due to Wnt stimulation were linked to genes which in general showed higher expression due to Wnt stimulation. So this in general, we think, means that these elements may be functioning as condition-dependent enhancers. So, and this in general gives some support to the idea that EMPRA elements are functioning in some ways similar to their function in the endogenous genome. Okay, so we also wanted to see if these active elements were enriched in any known genomic elements. So previous EMPRA studies have identified retrotransposons as enriched in EMPRA elements. So we wanted to check that as well in our data. So what are retrotransposons? These are elements that basically express themselves and then reintegrate into the genome. And retrotransposons have been hypothesized to contribute to the rapid evolution of the human brain. They've also been hypothesized to contribute to sort of copying different regions that regulate gene expression. So copying different enhancer elements throughout the genome. So ALU elements are one specific type of retrotransposon, and they occupy around 11% of the human genome. And the expansion of ALU elements has been specific in primates and has kind of co-evolved with brain expansion. So there's been some suggestion that ALU elements have overall contributed to brain size expansion and specifically cortical expansion. So we found that EMPRA active elements for our cortical structure associated library were highly enriched in ALU elements. So these here are bins of EMPRA activity, and here we're showing different types of ALU elements containing either the A or B boxes. These are regions of the ALU elements that function as promoters. And so those regions with both the A or B boxes in the forward orientation seem to have the highest enrichment for EMPRA activity. Interestingly, also, the EMPRA activity was specifically enriched in the left arm of the ALU elements, which contain these A or B boxes, and ALU elements that are the youngest, that have basically the least amount of mutations within them, showed the highest level of enrichment in these cortical structure associated EMPRA active elements. So what we think is that, what this implies is that younger ALU elements may have created these novel gene regulatory regions important for altering brain development and structure along the human lineage. To give some more evidence for this, variants within, EMPRA variants that lead to differences in activity of the EMPRA constructs were specifically enriched in these A or B boxes in the left arm of the ALU region. So this gives further support to the critical regulatory role of these specific regions in governing cortical structure and in ALU element activity. So strong ALU element-driven transcription may be an artifact of non-chromatinized sequences used in the episomal EMPRA library that are normally silenced in the endogenous genome. So we wanted to look for some functional evidence that these elements were actually functional within the endogenous genome. So we looked through a database of enhancer RNAs and upstream antisense RNAs that can kind of function to bring together enhancers and promoters in the genome. And we looked to see if our EMPRA active elements were enriched in these enhancer RNAs and upstream antisense RNAs measured in neural progenitor cells through a separate study. And we did, in fact, see some enrichment here, suggesting that these EMPRA active elements do exert some level of function in the endogenous genome, possibly such that they're serving as enhancer RNAs and causing promoters and enhancers to loop together. So cortical structure associated SNPs here were identified. Can we use all of this EMPRA activity to identify specific SNPs that seem to have gene regulatory effects, narrowing down our very large LD blocks here? So we found an individual locus that I introduced before associated with both the lingual and pericalcarine surface area. And we found multiple different SNPs where we detected EMPRA activity and allelic effects, and a subset of those where we detected actually condition-dependent effects. So in this particular variant here, we noticed that we see a very strong allelic effect at this location. And this allelic effect was actually blunted by Wnt stimulation here. So overall, we can try to suggest individual variants that seem to be leading to differences in cortical structure using the combination of GWAS and EMPRA. Okay, so actually, I'm going to skip this one for time reasons and just summarize here and then go into our iPSC work. So to summarize this, EMPRA and CAQTLs measure different aspects of gene regulation, and most cortical structure-associated loci contain elements with EMPRA activity and allelic effects, suggesting specific gene regulatory regions, SNPs, and stimulation states impacting brain structure at complicated loci. Also, EMPRA activity, at least in our hands, was largely driven by younger ALU elements, and this possibly suggests that enhancer RNA mechanisms regulating gene expression have contributed to human cortical evolution driven by these retrotransposons. Okay, so I have a limited time left, so I'm going to talk to you now about one of the last studies that we're working on. So, previously we've been focusing on using a population where genetic variation exists and then trying to walk along these different gene regulatory mechanisms, leading to explain differences in cortical structure. But an alternative approach is that we can take individuals where cortical structure was well measured and generate pluripotent stem cells from those individuals to study how well those pluripotent stem cells model the structure of the human brain. So we're doing this through a collaboration with what's called the Infant Brain Imaging Study, or IBIS. And so in this study, we're leveraging deeply phenotyped individuals from the IBIS study. They took high likelihood families where one child already was diagnosed with autism, and the second child was deeply phenotyped from infancy, from 6 months of age, 12 months, 24 months of age. MRIs were taken of the brain as the child grew up. So this is like a really unique data set because we have these longitudinal profiles of what the brain growth looks like in these individuals. So some of the families had a child with autism, and the next child was diagnosed with autism. We call those high likelihood ASD. Some had a child diagnosed with autism. The next child was not diagnosed with autism. We call those high likelihood negative. And then we have a control sample where nobody in the family was diagnosed with autism. So this is a very unique resource because it allows us to generate an iPSC population where we can enable the assessment of in vivo to in vitro relationships, as well as being able to study the cellular and molecular mechanisms leading to autism. So for this population that had undergone longitudinal infant brain growth trajectories, we generated iPSCs to kind of go back in time to an embryonic-like state and be able to understand what causes inter-individual differences in brain size. So this population provides us like a really unique opportunity to assess the fidelity of cortical organoids as a model system of the developing brain. So for this particular study, we took 18 participants in the IBIS study, each of which had undergone longitudinal MRI imaging from infancy. We drew blood to generate to gather peripheral blood mononuclear cells, reprogrammed erythroblasts into induced pluripotent stem cells, and differentiated them into cortical organoids. We acquired different measurements of those organoids, including phase contrast imaging, 3D imaging through tissue clearing, and single-cell RNA sequencing. And this project was done together with Rose Glass and Nana Matoba in my lab, as well as the IBIS crew, Joe Piven, Mark Shen, Jessica Giroux, and a stem cell core here, Adrienne Beltran. So here's just an image of what our cortical organoids look like with lots of nice neuroepithelial buds. And I think a lot of you guys have seen these before. So we found that when sampling about 200,000 cells across this population using single-cell RNA-seq across two different time points, we had day 14 and day 84 post-differentiation. These in vitro models of brain recapitulated the major in vivo cell classes of neurons, radioglia, cycling progenitor cells, and also cortical hem. We did find evidence for something I think Ron was indicating or implicating before, which is poor subtype specification. So these show the overlap between in vivo fetal brain, shown here from Arnold Kriegstein's data, and our subclusters from our cortical organoids. And in general, there's an overlap between, for example, neurons and our data. But there's not really good subtype specification, and similar with radioglia shown here. So, we also don't really get things like microglia or vasculature, which we kind of expected based on our differentiation protocol. So overall, we're generating some of the correct cell types. We wanted to then see if we get reproducible cell type proportions. So we evaluated this reproducibility because we generated up to 3 clones per donor in our study. So most iPSC studies are generating about, usually just 1 clone per donor due to the expense of organoid differentiation and iPSC reprogramming. But so here, using multiple clones per donor, we have the unique ability to determine how much genetics influences cell type composition, because we expect that multiple clones from the same participant will share genetics and be more similar to each other than clones across participants. So you can see here the cell type proportions at 2 different days, so day 14 here and day 84. And at day 14, we see very consistent cell type proportions regardless of any clone or donor. So just overall really, really high consistency. We actually didn't see any significant difference within participant versus across participant, likely due to just the fact that they're just so similar across each other. But at day 84, when more variability exists, we do see more similarity within participants than across participants, indicating that the genetic background of the donor is contributing to the differences in cell types that exist in that donor. So we then wanted to ask, do these organoid phenotypes relate to the cortical size of the participant from which they were derived? And we did this by measuring the organoid size, at least initially, as a simple phenotype, just the organoid cross-sectional area through microscopy. And we found that the trajectories of cross-sectional area are more similar within than across participant, which was encouraging. But then we correlated them to different phenotypes about the growth of the organoid as well as the growth of the individual from which they were derived. And we do see across these different donors that there is sort of a similarity between the growth rate of the organoid and the actual growth rate of the cortical surface area. So this suggests that organoid development is somewhat predictive of inter-individual variation. Next, we wanted to see not just the structure, but since we ran the single-cell RNA-seq, can we find individual genes within individual cell clusters that were correlated with differences in the brain size? We found one particular cluster which had around 3,000 genes that were associated with cortical surface area growth rate. This was particularly interesting because these genes were involved in cell cycle, something that we already predict based on the radial unit hypothesis is associated with differences in brain size, and were specific genes that were involved in microcephaly, had already been implicated in microcephaly, were also identified here as correlating with differences in brain size. We then were trying to understand what that particular cell cluster is doing. So we use what's called a pseudotime analysis to better understand the developmental trajectory of that subcluster. And so in that subcluster, it's like we have these radioglial cells that are deciding to form neurons or different types of neurons or radioglia. And right at the crux of that fate decision is where this single cell, this cluster 22, SC22, is found. So this cluster seems to be really important for determining fate decisions right at the point where you decide to form a progenitor or another neuron. And individual variability there seems to be predictive of what determines brain size later on in development. So to further demonstrate the relevance of these genes to brain size, we found that these genes that were associated with differences in brain size in our population were already previously associated, were enriched in those genes associated with microcephaly, as well as genes associated with risk for autism, developmental delay, and neurodevelopmental disorders. So overall, we think we've shown that there's some fidelity of cortical organoids for modeling inter-individual differences in brain size. And also, we've identified specific cell types and genes that may be important and contributing to this variability in brain size. So currently, we're expanding our approach to instead of just the 18 participants in the study, we've expanded to 115 different participants, which we're differentiating now. And we're acquiring different measures like single-cell RNA sequencing, lineage tracing, and light sheet microscopy to evaluate these in vivo and in vitro comparisons. Okay, so overall, genetic backgrounds influence cell type composition in organoids, as shown by higher similarity within multiple clones from the same participant. So using iPSC-derived human cortical organoids from deeply phenotyped participants with longitudinal MRIs during infancy, we identified in vitro phenotypes that predicted inter-individual differences in infant brain growth. We think this supports organoid fidelity. And finally, progenitor cell fate decisions seem to be, in early development, seem to be important for determining differences in cortical size. So overall, our lab is trying to sort of understand these mechanisms affecting human brain structure and ultimately function and leading to risk for psychiatric disorders. We do this using endogenous variation that exists in populations of cells, inducing variation through multiplex parallel reporter assays, as well as generating induced pluripotent stem cells to study the organoid fidelity. So hopefully using all of these different mechanisms, we'll be able to, all these different techniques, we'll be able to understand the mechanisms leading to differences in brain size and ultimately risk for psychiatric disorders. And so I wanted to thank individuals in my lab who have contributed to these. I've already sort of highlighted them throughout the talk, so I'll just pause here and ask for any questions.
Thanks so much, Jason. A lot of work here. Very, very interesting. So anybody, please jump in with questions. It's hard to know even where to start, but it's...
Hi, this is Flora. Hi.
Hey, Flora.
Hi, Jason. How are you?
Good.
Wonderful talk. I just had a relatively simple question. In the infant brain study, I think you showed some curve that charted the relationship between brain growth and likelihood of developing autism in the infant.
Yeah,
Yes,
this one.
that one, exactly. So is there a relationship between brain growth and diagnosis in the high likelihood?
Yes.
There is.
Yes, there is. At 6 to 12 months of age, there was found to be a cortical surface area hyperexpansion that leads to a brain growth, overall brain growth that's observed from 12 to 24 months. So
Okay.
basically, there is a hyperexpansion in the ASD group. And this predates diagnosis, which is generally at 24 months of age.
Right. Now, this is statistically significant with respect to the low likelihood or not?
Yes, yeah. And that's detailed in this paper here.
Yeah. Okay. And okay. And so I had another question about this, and I'm just yeah, and you're basically so in you're when you chose patients for organoid, how did you choose them? Was it unbiased or?
So yeah, so we tried to choose the overall goal was to try to get equal numbers in each of the three diagnostic groups, taking the individuals that had the most amount of sampling of the MRI measurements. So because not every, these were human studies, so sometimes the kids would come in for 6, but then skip the 12 and then would be 24. So we don't have for every subject every different time point.
Okay. Here is another question, which is a question for everybody, I think, not just for you.
Okay.
And here is the question. So macrocephaly is supposed to be slightly adaptive. That means it's slightly positively correlated with IQ. It's not a huge correlation, but it's there in general, in the population. So do you think that this macrocephaly in autism or higher rate of brain growth, whatever you want to call it, is that a different type of macrocephaly than what happens in normal? This is a question because often, as you well know, in our study, and I'm sure in those studies as well, this is a family trait.
Brain
So
sizes.
that means that the kids with autism have parents also that are macrocephalic. So suggesting that, it's not necessarily, maybe it's very similar, but on the other hand, it's also a maladaptive trait. So this is something that I don't think is currently explained by any of the published data, but I was curious to know what you think.
Yeah, it's an interesting question. I do think that the level of macrocephaly, at least in this population, which is a pre-selected population for having one sibling with autism, so they're already at high risk for developing autism. So in this population, that the degree of brain overgrowth was correlated with the degree of autism symptomatology. So basically, the worse, the bigger the brain, the worse the phenotypes were. So yeah, I also know, from our GWAS cortical structure that basically the same genetic variants that predispose you to a higher cortical surface area also predispose to higher IQ, as measured through GWAS.
Yeah.
So yeah, I agree that there's a both sides are being played here, right? So, and I
Yeah.
don't really understand why. Yeah,
Okay.
to be
Thank
honest.
you.
Yeah, I guess that was coming back to your question, Flora. Is it not a single axis of macrocephaly, but actually a different way to macrocephaly that is harder to control or harder to keep on track, and a different one? Or is it just simply a U-shaped curve where a lot of more is better until more is worse, and it's actually on the
I
same
think
trajectory?
I think the infant brain study should have measured parents as well.
Oh, that would have been, yeah. Well,
I don't know whether they did or not.
Yeah, my understanding is we don't have good information about the phenotypes in the parents. We do have information just about the individual, and occasionally we have, which we call the proband. Occasionally we have information about the sibling, but we don't have a lot of information about the parents. There is like some information, but I don't think it's a lot of information about the parents. Like you're basically asking for MRI measurements to know macrocephaly in the parents, right?
Yeah.
And that was not done. Yeah.
Yeah. Okay. Thanks.
Yeah. Thank you.
Yeah, and from the measurement of structure, there's no way to tell it's obviously the origin of any change in size.
Yeah, I don't think we would be able to sort of predict the sort of structure in the parents if that was the idea. I don't know.
Yeah, and by the way, I agree with the mechanisms you propose because we did measure parents. We did collect parents along with proband in our study. That was just an organoid study, no imaging.
Yeah.
But and so the differences in gene expression were actually computed versus parents, which often were also macrocephalic. So for sure, there are differences in organoid gene expression, but in vivo, in terms of what this macrocephaly actually is, what's the molecular cellular mechanism, whether that's similar or different, there must be some difference, undoubtedly, right? Is it a difference in extent, or is it a difference in quality,
Yeah.
in the process itself,
Right.
Yeah. I think it's a really good question. Yeah, I don't know the answer though. Yeah.
So, Key, did you have a question?
Yes. So Jason, for the organoid project, I also saw your preprint on this project. And there you also talk about association with the cortical ham population
Yeah.
with the later in vivo growth cortical rate, right?
Yeah.
And for me, I mean, I also was very excited that you actually used day 14 cultured organoid and do the transcriptomic, right? And then you are talking about cortical ham identities possibly associated with the cortical growth in vivo.
Mm.
And that's very interesting. And I think that time that you chose for the single transcriptomic is really ideal for exploring other regional identity. So I want to draw your attention to our previous work. Here, Nicola is on the call too. So we published a paper in Science 2023, single-cell transcriptomic data from macaque telencephalon collected from embryonic day 37. It started from 37.
Okay.
And we identified all the different brain organizer population and also regional identity of neuroepithelial cell early stage with the ventral, frontal, and occipital identity. And we also bioinformatically analyze the signaling interaction between these regional radioglial cells. And
Okay.
we talk about Wnt and BMP and specific molecules interaction involving this region, right? And I think if you correlate this 14 organoid data set with our the published data set, you might actually get more deeper resolution, not only the cortical hem, but also frontal organizer, right? And then
Yeah.
whether those things are actually related to the cortical patterning and then expansion, I think that could be very exciting to see.
Yeah, I think that'd be great because I mean we correlated to these kind of in vivo fetal from human, but like there's a limitation that we're mainly studying the cortex here. So, if we have cortical, so you said you have cortical hem from macaque?
Yeah.
Yes.
Hi, Jason. Hi, I'm Nicola Micali. So, by the way, Sulki is my wife. That's why she's talking
Oh,
about.
nice.
We are talking about the same paper. Yeah, I think it is. We published a data set where we profiled these different regions, some different organizers in the primate. I mean, the macaque. So maybe you can use the data set to basically define if there are other organizers in your organoids.
Okay.
Because the point, and maybe I mean, we also talk about this concept in our preprints in in a bioarchive, where basically it's possible that there is a correlation with the level of a Rosa Pattern Center or cortical hem, right? There is this balance in the expression of Rosa Pattern Center or hem genes, which may be correlated with autism.
Okay.
We saw that is imbalance in these cells in multiple ASD organoids. Actually, it was a collaboration with Flora Vaccarino, where we use their data set. So
Okay.
I also was interested to see if you look at if you switch slide, you go on the next slide here.
Here.
Yeah, I was in fact curious to understand if there is a variation in the cortical hem identity in cells expressing cortical hem signature.
The lightest yellow. The
Yeah.
lightest yellow.
Oh yeah, it's the bottom ones. Yeah, these guys are the cortical hems. Yeah, I
Right.
mean we used a organoid differentiation protocol that used cheer basically. So like sometimes we get these cysts. This was the Hangjun Song and Guoli Ming protocol is what we use for this particular differentiation. So yeah, so I think that's the reason that we're getting all of these. Like we have like a pretty high population of cortical hem, to be honest. And
Yeah,
I think
day
that
84, that's pretty surprising.
Yeah, but in fact, I mean, day 84 is quite surprising to see it is a ham population after so many days of differentiation. So I
25.
wonder if actually I have two questions related also to Sulki's point. How you define the cortical ham identity, and is it possible that actually in this protocol there are still undifferentiated cells which shouldn't be there?
I mean, it's definitely possible. Like, we
There's not
define
many
based on.
cycling.
Oh, sorry.
Right.
There's not many
It can-
cycling left in that. There's
Oh,
not that.
yeah,
Maybe
yeah.
the
orange is almost gone.
occipital cortical
Exactly. It's
identity.
possible actually that this is an occipital
It's not
identity,
the
not
ham
cortical ham
organizer.
identity.
Oh, so wait, so explain that. So why would it be? We did. It was basically based on like largely TTR expression was high in this cluster, So then we labeled as cortical hem.
Right. So basically, you use
Right.
just one marker to
Correlate.
basically
much.
identify
Oh, no,
the class.
I disagree. I don't think if they're TTR
Exactly. Right.
medialized, they're not visual cortex for sure. Some organoids have that. They
It's
have
more
an
likely
No,
to
I
be
understand,
choroid at that point,
Flora,
right?
but TTR is a no visual cortex for sure. But day 84 is still very early, and there anyway, TTR is a choroid plexus, is a cortical
Yes.
hem transition to choroid plexus. This is really not even a hem actually.
So
If you
depending
find
how deep you
TTR, you
want to
should
jump
also
into
find
this,
RSPONDY3 positive cells, right?
Jason, depending on how deep you want to jump into this with us, we have Nicola's macaque data, Flora's ASD organoid data with Alex Jordan, and the signatures of all the different organizers that Nicola and Soki are talking about live in the same analytical platform online. So
Okay.
if you wanted to look at all of these signatures in your data, and we could get your data online and not have it public yet, just to private to us, and we can look at all these things across all of our systems if you're interested in jumping in there.
And also, Flora, you guys see TTR, but at early time point. My
Yes.
argument is day 84 is a little bit too
No,
late.
I agree with that, but it is TTR does indicate hem-like, because the hem is right next to the choroid plexus.
Yeah.
It's not exactly, so it could be, Jason, that this is actually choroid plexus, not necessarily.
Exactly.
Oh, yeah.
It's like
I mean, maybe we
above.
should just label a
Yeah.
little bit more. Yeah. So
Also,
yeah,
ependymal cells may express TTR. I may be wrong here, but ependymal cells have several features similar to.
No, they're right next to each other. So the hem actually expresses a lot of Wnt3A and other Wnt genes.
Absolutely.
Very high.
The
And also
arspondins, yeah.
BMP and Wnt, essentially.
Right.
yeah. So maybe we should just
LMX1A, you should see that these clusters are LMX1A positive, RSPONDY3 positive, also TTR, but TTR is another state of the hem that we named cortical hem choroid plexus, right? Because so what I'm trying to say is that here this variation of the identity of the hem identity. It's interesting because it's showing, as we demonstrated also in our previous literature with Ron and Carlo, Michele et al. 2020 cell reports, that there is this variation in the state of these cells, right? Across different lines, there are different lines can express different level of these cell types. So this variation is actually makes sense. But day 84 is a little bit too late, in my opinion. So I wonder if, again, I told you, maybe you should check, not reconsider, check if the identity of these cells is really a cortical hem or choroid plexus, as Flora is suggesting, or ependymal, or again, occipital regions. So, I mean, which is probably unlikely, because if they are TTR, probably they are not occipital cortex. But
I mean, occipital cortex should be very distinct, should have very high level of FGFR3 and CUPTF1 or CUPTF2. So
but also RSPONDY3, which is basically is also a marker of the hem.
Oh, RSPONDY3.
Okay.
Yeah.
On the other hand, TTR is a transporter, which is clearly expressed in the choroid plexus. So I don't know about the hem. You can maybe do a subclustering of that cluster and see.
Yeah. Okay. These are good suggestions. Thanks. Yeah. Okay. So
we'll take a look. I have another question, Stein. So, Jason. So here, basically, you say that, but here there are autistic samples and controlled samples too.
Yeah. So it's only 4 autism samples. So we did not focus on the autism. Like we did not focus on diagnostic differences for this study, because we are like N equals. Yeah. I'll show you this. We just have 4 where they actually have autism, and then.
So you don't know if there is actually a propensity for the autistic cells to express a hem to give rise to hem cells.
We did not test.
Okay.
Yeah, I mean, I think basically we were waiting for diagnostic differences for this study because we didn't feel like we were powered to do the diagnostic differences. So this current study, we're just doing in vivo, in vitro. comparisons.
But you see some association with the cortical cluster size in vivo, right? With the
With,
cortical hem. So some
oh,
correlation
with cortical size. Yeah, with cortical size. So
right.
we compared cell type proportions of cortical size, but not across the different diagnostic groups. So we didn't do that like autism versus non neurotypical. We did not do that.
We found several genes expressed in the cortical hem in this bioarchive paper using Flora's the dataset from Jardon et al. We found several cortical hem genes deregulated in the autistic in ASD organoids. And also in the macaque paper, we showed also several genes which basically are associated with, usually are commonly associated to later expression in neuron, like MET or like actually, which are basically risk genes for autism. We found those genes in the cortical hem because we found we profiled so early, such early cells that now we could identify risk genes expressed very early.
Oh, cool. Okay. Very cool.
Okay, thank you.
Thank you.
Maybe I can just ask a question which is sort of related to this topic that we're discussing here. When you showed that image where you showed a very specific fate choice rather late in your talk, and you made you just went past it. Yeah. There's fabulous talk. So many things to discuss. So I don't mean to dominate at all, but it struck me that this was a very interesting kind of image here, and that it would be worth our while to think a little bit more about it. Many years ago now, a paper actually from Dan Geschwin's lab, maybe you were even an author on it, made an observation that in I think in autism brains, that the specification of differentiation was less acute. It was that the brains were less regionalized.
Yes.
Yeah.
Yes.
And it seems to me that comes up in this point here where you made the crucial fate decision. Is it that the fate decision is wrong, or is it that the fate decision is kind of degraded? It just isn't going through the fate decision with as much acuity or precision. It relates to our organizer discussion. That's why I thought it would be worthwhile to bring it up here.
Yeah. It's interesting. I mean, here we're not sort of focusing on diagnostic differences. We're just focusing on,
Right.
relationship to cortical size. So, whether sort of like lack of areal specificity, I don't think this is informative as to whether lack of areal specificity contributes to autism phenotypes.
No, but it's this kind of data that you'd want to have, right? Because this is a very high resolution kind of image about a fate choice.
Yeah.
Right. And
yeah,
I thought it was beautiful, right? Worth thinking about.
So, I mean, I guess this has driven some of our kind of ideas for the future, where now we're focusing on doing this type of lineage tracing, where we put in a barcode where all the progeny of a particular cell inherits that barcode. And that would allow us, hopefully, to, this is using pseudotime and a lot of statistical inference to understand what a cell is doing. But if we actually measure fate decisions, and, I guess in general, yes, I believe that alterations in fate decisions are going to definitely change the structure of your brain. And maybe those fate decisions are leading to differences in the cell types that are formed, such that, we can still have macrocephaly, like going back to Flora's question, you can lead to increased proliferation to get macrocephaly, but what cell types are comprising that macrocephaly? Is it, too many upper layer neurons, not enough lower layer neurons, the opposite? I don't know. Like maybe that's what leads to macrocephaly but autism phenotypes versus macrocephaly and high IQ. I think those type of things we can't really see with this resolution yet.
Well, we had an hypothesis in Alex Jourdan's paper And that was that there was a disconnection between preplate, the choice between preplate and dorsal cortical plate in radial glial cells. That was crucial to, which is not exactly identical to what you're saying, but related to that, right? Because either cells become radial glia and enter asymmetric division, or they keep being younger neuroepithelial cells, right? And maybe they differentiate into preplate, but that decision to generate the cortical plate is delayed. So that was our hypothesis, but you know, I mean, this is just one. And you know, plus we differentiated the two type of ASD, the normocephalic and the macrocephalic. It would be fantastic to correlate that with in vivo data. In fact, if you already have IPS cells from this longitudinal imaging cohort, do you have normocephalic ASD as well as macrocephalic?
Yes. Yeah, I mean, you can see in like there's not huge macrocephaly even in this population, so it's hard to see the individual donors, but there's definitely normocephalic like individuals in the ASD group as well. It's just on average that there's a cortical surface area hyperexpansion.
So that would be interesting to see, to look at
to separate those, the normocephalic ASD, the macrocephalic ASD.
Or at least do some sort of correlation, phenotypic correlation with that.
Yeah, agreed. Yeah.
I just make a comment about the part of your talk where you talked about regulatory elements.
Yeah.
And there's here again, there's a huge number of things to think through. Thanks for giving us such a comprehensive summary of what you're doing.
Sure. Yeah. Sorry.
It's very striking at the moment that I'm thinking of labs from the East Coast. Ryan Toohey is a senior author on a paper recently in Nature, and Jay Shendur has similar papers, slightly different approaches, where they're doing genome-wide analysis of regulatory elements, CREs.
Yep.
And here you chose, I'm just trying to remind myself and remind the audience of what you did. You chose specific regions that you thought were particularly interesting and explored their phenotypes. Is that right?
Well, we chose every genome-wide significant locus associated with inter-individual differences in cortical surface area or thickness. So, I mean, I guess you could, we only chose those that were genome-wide significant, and then we took all the SNPs at that locus, so all the SNPs at that locus, and put them into the emperor constructs. So, whereas, I think the ones you're talking about, like they used ENCODE data to take every cis-regulatory elements and then input them into the emperor libraries. Yeah. So
and they use slightly different vectors in the two approaches, but
so ours
but I'm just sort of making the point that we're coming to a stage here where we're really going to have extremely powerful genome-wide insight into the functional effects of non-coding regions. And right now, they're not using brain cells in these studies, but they're getting very nice cell type differences by using cell lines. And so there's really going to be an explosion of our understanding of this kind of control.
Yes. Yeah, I think that's very true. I think, like you said, they found very big differences in regulatory activity within different cell types, which to me totally makes sense because you have different transcription factors that are acting within these different cell types. We also used, for example, like Wnt stimulation or lack of Wnt stimulation, and we see some level of difference. There's not huge differences due to Wnt stimulation, but we see some level of differences. Again, probably because transcription factors are being recruited to the nucleus due to the stimulation. So
maybe I can take advantage of your discussion here. The thing that about Ryan Toohey's group, there's also a guy at Yale actually who just blanking on his name, who's a senior author on this paper, but is they say that actually their synthetic sequences show much more cell type specificity than than the endogenous genomic sequences. And I think that's an extremely interesting finding, actually. And whether you might find that here as well, that actually that these sequences that are risk sequences are actually more specific, rather in contrast to what we were saying earlier about a lack of regional specificity being a risk factor.
I mean, it would be really cool. So I think you're suggesting to like do this in different cell types, like to use the
Yeah.
same library, but in different cell types or different stimulation states that are non-brain related. Is that
Well, I guess what I'm saying is I think these
Regionally
sorts of
defined.
issues really need like careful thought because there's a huge amount of work goes into it.
Yeah.
But it's extremely interesting to think through what it is we really need to know. And I think normally we think of. I think what I'm trying to say here is that the endogenous system is going to be balanced, right? That's the conclusion I draw from the Ryan Tui Nature paper. The endogenous system is balanced because the genes are doing different things in different places. But if you ask for synthetic sequences that show cell type specific effects, actually those synthetic sequences are far more cell type specific than actually you find in nature. That strikes me as being an extremely interesting finding.
Yeah.
Okay.
Yeah.
Peace.
Agreed.
Thank you again. Yeah.
Thanks, man.
Anybody else with any more questions? Well, thanks for coming. And as you can see, Jason, we're interested in so many different facets of
Thanks.
what you're doing.
Yeah,
and
thanks
for
so
sure, I'll
much.
reach out and poke you again about the notion of getting some of these data sets and gene lists about regionality and organizers in the same space.
Yeah, that sounds great. Yeah, we put on to NDA, NIMH Data Archive, the raw reads, but
Okay.
maybe the like
Yeah, the process data would be great to have out too. That's
Process.
great.
So on Nemo, that's what you have is
Yeah, so there's Nemo Archive, which is more like the raw read archive, but then there's
Okay.
Nemo Analytics, which is what I was talking about, which we use almost 100% process data. And like I said, the macaque atlas that Nicola and Silky worked on is there. The organoids from Flora and Alex are on there. And then we've also extracted markers from each one of Nicola's monkey organizers, which we can project into your data, for instance. So we can see the level of the hem signal or the rostral patterning center in your individual cells, in your individual organoids. So we can make kind of what you made with those proportion plots, but for signals of the different organizers that are not just the hem, but a whole bunch of I think it's, what is it, Nicola, like 10 or 12 organizers we got markers for out of your data. Oh, he dropped off. Wasn't it so?
Yeah, there's multiple organizer state.
Okay.
But
Yeah.
yeah, I'll bug in. I can show you what we have there and how it works.
Okay, yeah, that'd be great. Yeah, I think that would be great. Yeah, especially that definitely not super familiar with as familiar with the hem choreplexus.
Yeah, for
separation.
organizer state, that's the most powerful data set, our data set, because human data set hasn't been collected that early. It's because it was macaque, it could be collected early.
I
So
see.
it's much more robust cell fate than you can actually explore.
I
So
see.
that's why I think it's perfect with the day 14 organoid. Normally people
Right.
haven't really explored that early
Yeah, I think
organoid,
all of
right?
us were quietly happy when we saw the early time point that you had taken there. It's rare.
Oh, nice.
People want the big mature neurons, right? Hundreds
Yeah.
of days later.
I mean, honestly, we were surprised that we were getting mainly the in vivo in vitro correlations at the earliest time point, at the day 14, not at the day 84. So like, yeah,
That's
we
very
were
cool.
Right.
we did not
That
expect
was
that.
also very cool. Yeah.
Yeah.
Very cool.
Mhm.
Right. You get the progenitor readout, not the.
Right. Super early progenitor readout. Yeah.
Awesome. Thanks again, Jason.
Thank you so
You'll
much, Carlo.
definitely
Thank you.
be looking forward
Okay.
to continued interacting. Thanks again.
Okay. Yeah. Thanks so much. Yeah. Take care. Talk to you all later. Bye.
Bye.